we have been working together since the 1980s. Back in 1975, I organised the second meeting on proteins of iron storage and transport in Louvain-la-Neuve, and out of that came a collaboration with Hoechst, who were trying to develop a one-off drug, orally active, that would be able to deal with the anemia of pregnancy. Women who have been pregnant and been anemic will recognise the syndromes. When I was writing my book on iron metabolism in Whitstable, one of the ladies who frequented the two brewers every lunchtime said, "Bob, it's like shitting iron bricks." And I think you get the message. Something that would be less constipative than ferrous sulfate was a highly desirable product. And they discovered this ferrocene derivative. So you've got two aromatic rings with an iron stuck between them. And in Mr. Rat, that goes across the gut, straight to the liver. The animals that are anemic, with one single dose of this stuff, their proteohemoglobin is back to normal. It works exactly the same in dogs. When they tested it in the Wiesbadener Klinikum, half of it went out in the urine in the first 24 hours and the rest in the second 24 hours. It is not resolved in man. But it turned out to be a brilliant way of loading iron animals and making an animal model of iron overload. And that's where David comes in, because we then collaborated with him to show that some of the iron didn't just go to the liver and the spleen, it also went into the brain, which meant that we actually had a model for loading the brain. And as you will hear, one of the characteristics of Parkinson's is accumulation, which was first shown by David, is the accumulation of iron in the substantia nigra. And that iron, if we can remove it, could be a target for therapeutic treatment of Parkinson's. So we actually showed in an animal model of Parkinson's that iron chelation therapy will improve the condition by allowing the key enzyme, which is defective in Parkinson's, tyrosine hydroxylase, to be increased back to normal values by treatment with chelators. And that led, as you'll hear, to clinical trials with iron chelation therapy in patients. But all of that is part of the story that David will tell you. I'm not going to stand in the way anymore. I'm very pleased to have you here, David, and I have explained to you if I disappear abruptly, it's because I might require to make a pit stop downstairs.
Have you got the clicker? It's on there. Right, okay. Right, let me just start. Yeah, right. Okay, so currently I'm based at Parkinson's UK. So previously I was Deputy Head of Division of Brain Sciences at Imperial College. So I was at Imperial College for 24 years, and then joined Parkinson's UK. Now I'm the Research Director. So just a little bit about Parkinson's UK. So it's a membership-based charity. So we've got about 37,000 members, most of which have Parkinson's. It's been going about 54 years, and we're the largest charitable funder of Parkinson's research in Europe. So currently we've got 37 active projects, probably about 33 million euros in investment. So 13 of those projects are part of what we call our virtual biotech. So we have more of a commercial arm where we put seed money into projects to take them to the next stage, and then we'll partner with pharmaceutical companies later on. So 6 of those are in preclinical phase, 6 phase 2 and 1, which I'll talk about is a phase 3 trial. So Parkinson's itself, I'll start off quite gently here. So we've got about 145,000 people in the UK, and about 8 to 10 million people worldwide have got Parkinson's. So age is the biggest risk factor, and as you can see here, as you go through the decades, you can see that the incidence of Parkinson's disease increases. So it's the fastest growing neurodegenerative condition. So it's overtaken Alzheimer's, and so within the next 35 years there's potential for a doubling of the incidence of Parkinson's. And I set up the Parkinson's UK Brain Bank at Imperial in 2002, and if you look at the healthy control brains which are coming in, 7% of those people have the early signs of Parkinson's, but it hasn't progressed to a stage where they'll get clinical symptoms. So if you think about it, there is a whole pool of people who could potentially develop Parkinson's, and as we treat heart disease, cancer, and more successful at keeping people living longer, more and more people are going to develop Parkinson's. So 20% of those are diagnosed under the age of 50, and about 10% of Parkinson's has a genetic origin, and I'll talk about that more later on. So it's more prevalent in males than females, depending on which literature you read. It's just over 2 times in some studies. All ethnicities are affected, but for some of the limited studies that have been carried out in black and in certain Asian communities, there is a slightly lower prevalence of Parkinson's. So it's classically known as what's called a motor syndrome. So you get deficits of movement, so that you get a resting tremor. You'll have rigidity in the limbs, the trunk and the neck, and also you have what's called akinesia, or slowness of movement. So they're slow to initiate movement, and often you'll get a problem with posture and balance. So that's the classical clinical symptoms, but there are over 40 other different symptoms that can affect somebody with Parkinson's, and it's mainly these. Sorry about the formatting, it's not come out well into the PowerPoint, so it's chopped some of the things up. So these are what's called the non-motor symptoms of Parkinson's. So within the brain, there are also changes which affect areas that lead to depression, anxiety. It also affects the sleep centers within the brain, and there is a syndrome called REM sleep behavior disorder, and if people develop that, they've got a 70% conversion rate into Parkinson's. It affects your visual systems. It also affects the autonomic nervous system. You have difficulties controlling your blood pressure. Affects urinary function, sexual dysfunction as well. So you can see it's a wide-ranging disorder, and it's often these non-motor symptoms which there are very little treatments for, that are more troublesome for people with Parkinson's than the motor syndrome. Yeah, unfortunately it's just chopped. This is why I wanted to run it on my own computer. But so this is all about pathology. So within the substantia nigra, so this is the source of the main dopamine pathways which control our movement. So dopamine is acting almost like the oil in your engine, and so without the oil in the engine, your engine is going to seize, and so you have problems with movement. Now dopamine, when it's metabolized, will actually get to neuromelanin, which is this peppered granule substance here. So when you slice through the brain at the level of what's called the substantia nigra, so you can see pigmented cells, and this is a healthy brain here. So you can see a dark line of pigmented cells. And in the Parkinson's case, you can see that this has become depigmentated. So the cells have died in that area. So that's the classical area for the diagnosis. And one of the other pathological features is the Lewy body. So this is this stained body here with this pale halo around it here. And then later on it was found that these Lewy bodies contained a protein called alpha-synuclein, and here you can see the immunostain. So it's not only in the Lewy bodies, but you get these thread-like structures which are called Lewy neurites. So these are the processes of the neurons which stain for alpha-synuclein. And so it's a spreading condition. So it starts off in the lower brainstem at the dorsal motor nucleus vagus, and in fact some people say but it actually starts in the gut and actually progresses up the vagus to the dorsal motor nucleus of vagus, and then starts to spread up the brainstem. So at this point, what's called Braak stage 1 and 2, this is before anybody is showing any symptoms of Parkinson's. So this period could be, up to a period of about 10 years or even more in some cases. And then once it starts to get to the substantia nigra and causes the dopamine loss, then you'll see clinical symptoms after you've lost about 60 to 70% of the neuron. And then from there, it'll actually start to spread into other areas of the brain. So the protein, which is called alpha-synuclein, it's like all of the neurodegenerative conditions. They all have a bad protein which misbehaves. So in Alzheimer's, it's your beta amyloid and your tau, and in Parkinson's, it's the alpha-synuclein protein. So it normally exists as a monomer or a tetramer, and it has a really wide-ranging function in vesicular trafficking. So it helps the snare protein, latch the vesicle onto the membrane, and then you can discharge the neurotransmitter into the synaptic cleft. So it also has a role in stabilizing the electron transport chain in the mitochondria, which are the powerhouses of the cell and producing ATP. Unfortunately, when alpha-synuclein, because it's an amyloid type protein, has a propensity to misfold, and when it does, it starts to play up and mess up lots of different cellular processes within the cell. So normally your proteasome gets rid of bad proteins, but the alpha-synuclein, misfolded alpha-synuclein, inhibits the proteasome. It also inhibits complex 1 in your mitochondria, blocking ATP production, and so you get spill-off of free radicals, the superoxide radical, from the mitochondria. It also interferes with the autophagosome, so you're not able to get rid of defective cellular structures. So it causes havoc within inside the cells, and then the cells gradually die. Now, I've probably put a bit more of a controversial title, word in my title about prevention. So recently, there's been a new biomarker for the diagnosis of Parkinson's. So this is called the alpha-synuclein seeding assay. So if you can look at the sort of the pinky purple line here, so this is where you get native alpha-synuclein. You put it in a shaking platform, and it's shaking for hours and hours and hours, and eventually the alpha-synuclein will start to aggregate together. But in a normal individual, if you put some normal CSF, cerebrospinal fluid, in there, this takes ages. But if you take a cerebrospinal fluid sample from somebody who has Parkinson's, or somebody who's got a risk of developing Parkinson's, and they have this misfolded alpha-synuclein in, if you put it into this reaction, it starts to cause an aggregation very quickly. So this bad protein acts as almost like a prion seed in the reaction, and then it starts to aggregate much quicker. So this now is a new assay for detecting Parkinson's, and so you can actually pick up people many years before they're even diagnosed. And what you can do, so that's about 87.7% accurate, whereas if you combine it with the University of Pennsylvania smell test, you can do this as a screen to start with, because again Parkinson's affects the sense of smell, and this disappears quite early on. So you can screen and then test the lower 15th percentile of those people with a really bad smell deficit. You couple it with the alpha-synuclein seeding assay, and it becomes almost 98% accurate there. So then you can use the DAT scans. So this is a measure of the terminal density at the end of the dopaminergic neuron. So here you can see in a healthy individual, the hotter the colour, the whiter the colour here, the more nerve endings there is. And you can see in the Parkinson's cases, this has degenerated quite a lot already. So then you can start to measure with time, with a DAT scan, the loss of the dopamine function within the system. So these are some of the genes that have been implicated in Parkinson's. So the SNCA gene is the one encoding for alpha-synuclein. So there are 3 gene mutations within the alpha-synuclein gene itself, and if you carry those gene mutations, you're very likely to develop Parkinson's. So these gene mutations cause the protein to misfold even more likely than somebody without these gene mutations. Then there are some, so this is a gene I'll talk about later on, this is LRKK2. So this gene, again, for some of the forms of the LRK2 gene mutation, if you're a carrier of these, you're very highly likely to develop Parkinson's. So then there are genes called PINK1 and Parkin, which are again rarer gene mutations. So all in all, these gene mutations are probably accounting for about 10% of those people diagnosed with Parkinson's. So if you carry one of these gene mutations, it's got what's called a high penetrance. You're more likely to actually develop Parkinson's than not. But there are some genes which really just increase your risk. One of the forms of the LRK2 mutation, which is the most common, which is the G2019S, and also the GBA gene, I'll just call it now. I've come back to that one. So if you have these genes, basically they increase your risk. If you have them, you don't necessarily develop Parkinson's. It's not in every case, but it increases your risk of developing Parkinson's. So then probably most of you have heard of GWAS studies, the genome-wide association studies that have been carried out in many of the disorders, and multiple risk genes have been identified. So exactly how these work, we don't know fully yet, because quite a lot of these SNPs or these gene changes are actually in the non-coding regions. So we don't know exactly what genes they're controlling at the moment. So there is quite a big genetic component. 10% of people have Parkinson's through a gene mutation, but then there are some risk genes which enter the equation as well. So this is just a diagram from a review that we did many years ago. So this is some of the sort of neurodegenerative processes that occur in sporadic or non-genetic forms of Parkinson's. So way back in the sort of early 90s, we showed that there was a complex 1 deficiency in the mitochondria. So this can produce oxidative stress. And as Bob mentioned earlier on, during my PhD, which was also funded by Parkinson's UK, so I've had a long association with the charity. So we showed that there was an increase in iron that could cause oxidative stress, and again it can interfere with protein processing. So the proteasome is very inactive in people with Parkinson's. So if you produce bad proteins, there's not many ways of actually getting rid of them outside the cell. And as you can see here, you've got bits of DNA, and this is to indicate where the gene mutations map onto the known processes that are present in sporadic Parkinson's as well. And also you've got inflammation down here, which plays a role as well. So misfolded alpha-synuclein, when it comes out of the cells, can actually stimulate microglial infiltration and activation. So then you'll get cytokines coming in, and inflammation takes a big role in the disorder as well. So the standard symptomatic therapies are all around dopamine replacement. You can't give dopamine itself, so you give the precursor L-DOPA, and this is rapidly converted by the dopa decarboxylase enzyme to dopamine. So that's packaged and released via the normal mechanisms. So these drugs are fairly effective during the first few years, but then you need to increase the dose because you've got fewer and fewer dopaminergic neurons to actually formulate the dopamine from the L-DOPA and release it. So the second wave of drug development was all about trying to create agonists which work directly on the postsynaptic dopamine receptors. And then the third phase of drug development was, we're producing all this dopamine, why not block its breakdown so more of it can go back into the storage sites again and be released later on. So we've basically got a wave of drug development in this area. Although these symptomatic therapies are quite good, they don't slow the degeneration down at all. They're associated with many side effects. You can get dyskinesias, which are abnormal movements. So really the sort of the holy grail is to develop drugs which actually slow Parkinson's down. So this is Kevin McFarlane. He's an ex-scientist and he's developed Parkinson's. But what he generates in the Journal of Parkinson's is what's called the Hope List, and he does this analysis every year. So he looks through the different clinical trials websites to find out what clinical trials are happening in Parkinson's. And as you can see from this dartboard picture here, that there is a huge number of drugs coming through the drug discovery pipeline. So the bottom part of the dartboard is for symptomatic therapies. So not only about improving movement, but there's also things for preventing falls. There's things in there for improving, alleviating constipation, alleviating anxiety and things. And in the top part, it's really where we're mainly going to be focusing the rest of the talk is what we call disease modification. So these are for actually slowing the disease down. So as you know, in the clinical trial space, so phase 1 is where it's mainly looking for the safety. So these are the first into man trials, and then as you get closer into phase 3, so these are taking the drug closer to clinical use. And so there are a number of drugs now which are in this phase 3, and I'll talk about another drug which is just entering that area. So it is quite an exciting time for Parkinson's research. So if alpha-synuclein is one of the primary culprits of causing the disease, quite a lot of the therapies are actually focusing on targeting alpha-synuclein. So as we age, we accumulate and make more alpha-synuclein, and age is one of the big risk factors. So you've got people who have a duplication or a triplication of alpha-synuclein genes, so they're more likely to develop Parkinson's. So obviously, if you can try and cut down the amount of alpha-synuclein, and that's what some of the approaches are actually looking at RNA interference, but then you've got the potential of taking it away from its normal role within the body. So how much can you knock it down without really interfering with its normal function? So a lot of drugs approaches are concentrating further down the pathway. So you can look at drugs which prevent or limit the downstream consequences of the alpha-synuclein accumulation, or you can develop drugs which either actually stabilize the alpha-synuclein or disaggregate it once it's started to misfold and aggregate together. One of the other approaches is that since the cell knows that alpha-synuclein is toxic in its misfolded form, as it starts to accumulate, the cell will then start to kick it out of harm's way. But then, unfortunately, what can happen is this can be taken up by neighbouring cells, and then it starts to propagate the process throughout this prion-like type of mechanism. So there are some companies which are working via an antibody-mediated mechanism. So I'll just talk about the first drug, this is Anali 138B. So you get the alpha-synuclein monomers, and then they misfold and start to aggregate. And so they go through this oligomeric period where these are the real toxic components. So these oligomers then go on to form fibrils, and then these fibrils then clump together and they start to form Lewy bodies. So what Anali 138B does, it takes these oligomers and then it actually fosters their conversion back into the monomeric form. So this was actually discovered in a small screen. They only looked at 20,000 compounds, and they came across anilin 138B, which was shown to actually disaggregate or take the oligomeric form and actually convert it back into the monomeric form. Yeah, so all my titles have gone. So, so this is just a pyrazole compound, and it has been shown to suppress the oligomer formation in vitro and in vivo. And it's inhibiting the pathogenic protein accumulation and neurodegeneration. So it improves the survival time of mice which have got the models of alpha-synuclein overload. And also in other models of prion disease, it also inhibits the formation of the toxic forms of those prions as well. So in transgenic animals where you're overexpressing alpha-synuclein or overexpressing the mutant forms, normally these go on and produce a similar Parkinson's type model. And in these models, it's been shown to reduce the protein deposition and actually improve dopaminergic function. So this is even if you start the treatment later on in the course of the animal model when they're starting to show deficits, and you add in the treatment then, these, the anilin 138B was still shown to be effective even at this stage. So that would be mirroring, adding this drug into people who have clinical Parkinson's now. So in December 2019, MODAG began the first phase 1 study in healthy volunteers, and then this has moved on to a phase 1B in 48 Parkinson's subjects, and this was shown to be safe even at fairly high doses. And then this has actually gone into a phase 2 now, where they're looking at safety and efficacy. Because Parkinson's progresses quite slowly, these clinical studies in Parkinson's have to be run for quite a period of time, and so it makes Parkinson's clinical trials very expensive. So the other approach, which is by Roche, is an antibody approach, so that as the bad protein, the alpha-synuclein, is kicked out of the neuron, the antibody that you're actually infusing into the subjects will actually capture the alpha-synuclein from the extracellular environment and take it away and denature it and get rid of it. So in the phase 1 studies, they showed that as you escalate the dose of the antibody, you can see a decrease in the circulating alpha-synuclein pool there. And so this went on into a phase 2 study, but unfortunately it failed its primary endpoint. So they were looking for a significant change in the rating scale. So this is the Unified Parkinson's Disease Rating Scale, the UPDRS. But what they looked at was the part 3, which is specifically looking at motor function within Parkinson's, and that you could see that there looks like there was hints of having some effect at the later stages within the trial. And if you look at the digital data, and this is where digital devices which can capture movement every day in a subject are starting to come into play on their own really, you can see a significant difference. But since these haven't been approved by the regulators for in clinical trials, these are still secondary measures. You can't use them as a primary outcome measure. So with this tantalizing thought that you might be seeing something at the later stages, plus some changes in the digital assessment, they've actually carried on with this approach. And this is really riding on the back of quite a lot of the antibody therapies which are coming through as effective in Alzheimer's. So this is where the first sort of gene mutation that we'll talk about. So this is the leucine-rich repeat kinase, or LRRK2. So in the brain in healthy individuals, it's really not very prominent at all. So you've got relatively low expression of this in the healthy brain, and if you knock out the LRRK2 altogether, it doesn't induce Parkinson's at all. It does, however, have quite a significant role in health in the periphery. It's essential for kidney function, lung function, and also in immune function. So straight away you can see that if you're trying to inhibit this kinase, that you might have some problems with off-target effects from these drugs. Whereas in Parkinson's, and particularly in those people who are carrying the gene mutations in the G2019S form of LARC, you get a marked increase in the kinase activity. And so you can actually start to see cytokine increased expression in Parkinson's. You get problems with infection control. And these cytokines can actually then lead to an increase in permeabilization of the blood-brain barrier, and you'll get the influx of immune cells within the brain, and this will cause a heightened immune response within the brain. So also, particularly in the microglia, you get an increase in activation with the LRRK2 mutations, and this starts to get an increased expression and activation of microglia. So the first drugs to target this are really starting to come through fairly recently. So this is the sort of the nice diagram about LRRK2. So it's a kinase, and you have this classical rock core kinase motif here, and here you can see the molecular structure of the kinase and the binding pocket here. So these kinase inhibitors sit at the key pocket site here and inhibit activity. So there's now a wealth of kinase inhibitors that are being synthesized and actually going through preclinical and clinical studies. So there are many companies now within this space. So some of them are still in the preclinical phase, while other ones are going first into man. The leading molecule was initially discovered by Denali, who then went into partnership with Biogen. And so this is moving into a phase 2, phase 3 study, and that's the molecular structure of DNLL 151. So there is a crossover here. So the idea is with these drugs that have specifically been focusing on these gene mutations, is that you may see benefit in sporadic forms of Parkinson's. So there is an increase in LRRK2 activity in sporadic or idiopathic Parkinson's. And within the G2019S mutations, so these carriers have an induced mitochondrial damage there, and these kinase inhibitors will actually restore mitochondrial integrity in Parkinson's. But there is that problem that within the high doses, when they went into primates looking for toxicity, they did see some significant inhibition of lung function. So then it's really concern about whether this translates into man, or is something that you specifically see in the higher primates. So Denali have gone into these phase 1 and phase 2 studies. So first of all, they were looking at multiple ascending doses over 28 days, and then they went into a double-blind, placebo-controlled study in Parkinson's patients, looking at 3 different doses. So in all of these studies, it was found to be safe, well tolerated, and they really didn't see any major changes that were worrying in lung function and renal function. But again, these are fairly short dosing studies, but you've still got to have in your mind about, is this long term going to have these side effects? So I think there is a bit of uncertainty in the field, but it's not really stopping people develop these drugs, or whether you can actually develop an inhibitor that would just work in the brain and not have those CNS effects. So it's gone into the phase 2, phase 3 studies now, and they're actually looking at both sporadic Parkinson's and people carrying the LOCK2 mutations. So this is glucose ceramide beta 1 gene. So this is the GBA1 gene, which I'm having difficulty saying this evening. So it encodes for a lysosomal membrane protein that cleaves beta glucidic linkages in glucoceramide. So basically, it's really necessary for the normal function of GCase. And GCase has a very important role within the lysosome about getting rid of bad proteins. So alpha-synuclein is a classic bad protein in Parkinson's. Normally, your lysosomes should actually get rid of the alpha-synuclein and degrade it. But in people that carry the GBA1 mutation, their GCase is either defective or it's decreased in activity. And so if you have a decrease in GCase activity, then the bad proteins build up, and so then you will get alpha-synuclein aggregation. So defects in this gene have been shown to pose a major risk for Parkinson's. So GBA mutations classically cause Gaucher's disease, which is a lysosomal storage disease. So people who carry these gene mutations have got a marked increase in risk of developing Parkinson's, but it's not a given that they will actually develop Parkinson's. So there's a whole host of different ways in which the GCase really acts here. So not only is it increasing your formation of toxic oligomers and Lewy bodies within the disease, but the lack of GCase really does impact on mitochondrial function, also causes an imbalance in lipid metabolism, creates ER stress, and also decreases your ability through the autophagosome to actually get rid of defective cellular organelles, and also activates some of the inflammation pathways via the NLRP3 processes. So there's a number of approaches that are looking at trying to correct for the GBA mutation. Obviously gene therapy, can you replace defective genes with the correct gene? So they're looking at enzyme replacement therapy, substrate reduction, but one of the approaches that I'm talking about now, which is a chaperone process. So if you can actually guide the GCase or modulate it or sort of nurture it back into its right shape, it will have its normal function. So there's quite a lot of these chaperone effects as well that are being studied. And this is a drug which is going into phase 3 again. The titles have just, I shouldn't have converted the anyway. So this is ambroxol again. So this was from a screen. So they looked for compounds which could improve GCase function with inside cells and protect them against the misfolded alpha-synuclein. And so ambroxol has been in this cough mixture, so it breaks down phlegm, so it gets rid of congestion within the lungs, and it's been used in medicine for many, many years, very safe. And so in an open label phase 2 study, it was shown to have quite a good effect on Parkinson's symptoms. And also it was well tolerated. So a consortium of funders have just given the go-ahead to start a phase 3 study. So this is going to be in 360 Parkinson's subjects. 50% are the GBA carriers with Parkinson's, and 50% again are sporadic Parkinson's. So this is going to be a randomized placebo-controlled study, 12 recruitment sites in the UK and 2 in Israel. And this is because the Ashkenazi Jewish population have a higher incidence of GBA mutations. So having recruitment centres out there means that you can recruit quite well people carrying the GBA mutations. So it's going to be 2 years on drug with an open label extension. And so this was a consortium funded at 5.7 million. So that is just going through setup at the moment. So hopefully it should start before the end of the year. So this is what really got me interested in Parkinson's, and that was the increase in iron, which I showed during my PhD. I was very fortunate to pick a project that really did lead to some major advances. So I was using a technique called inductively coupled plasma spectroscopy. So I was working in a geology lab. The guy there was analysing his rock samples for gold, and then I was using his machine for analysing brain samples, measuring iron. And so it was the first conclusive proof that there was an increase in the Parkinson's brain. But now there are several different techniques. Thankfully you don't need to mince so much brain up to find the iron. You can use a sophisticated MRI scan, and here's your Niagara here. And as you can see from the control to the Parkinson, you can see an increase in the Niagara. So it's almost like you've got the eyes and almost like the ears looking at you. That's where you could see the increase in iron there. So what's happening with this increase in iron? So in the early stages of Parkinson's, because there are fewer and fewer dopaminergic neurons, so really the neurons need to rely on their synthesis, so they increase synthesis of the neurotransmitters, so you need more of the synthetic enzymes, and those enzymes need iron as a cofactor, and I'll come back to that later on. But as the disease progresses and you accumulate more iron, it can have the potential of generating reactive oxygen species, and as you know, they're bad news, particularly for the brain. So there are 3 iron chelators which were in clinical use, and in the study that Bob was talking about, where we developed the model of iron overload, we showed that iron chelators could actually go in and take the iron out of the brain. So then we tried it in a model of Parkinson's, and this is what's called the 6-hydroxydopamine model. So this is a toxic breakdown product of dopamine itself, and so if you inject it in high concentrations into the pathway, it will cause it to degenerate. So if you look at the 6-hydroxydopamine treated animals alone, you cause a partial lesion of about a loss of 45% of the dopaminergic neurons, and this is mirrored by a loss of dopamine of about 65%. So if you give the iron chelators, you can see that if you're dropping down in these bars, they're all proving to be quite neuroprotective. And deferoprone, which is the smallest molecule, so each molecule of iron will involve binding from 3 molecules of deferoprone. This gets across the blood-brain barrier very easily, and we could show that it reduced down the neuronal loss in the animal model, and almost reduced the loss of dopamine almost down to the controls. So desferosox, which is produced by Novartis, is also active, and also desferrioxamine, which is a very large molecule, thought not to cross the blood-brain barrier, actually does get across in small amounts, and as you can see, it did decrease the neurodegeneration there. So together with Bob and Bobby and a few other people in Imperial, we did a phase 2 safety and tolerability study of the iron chelator deferoprone in Parkinson's, and then at a similar time, David Devos's group in Lille also showed that in animal models, the iron chelator deferoprone was neuroprotective, and in a small phase 2 study, here you can see the MRI signal here. So it's an R2 star MRI sequence, and as you increase with time, you can see the amount of iron being drawn out of the brain in the Parkinson's cases that were treated with deferoprone. And so this was mirrored by, if you're dropping below the line, it's an improvement. So the drug treated are the black bars and the clear bars were the placebo, which you would expect to get worse over a period of time. And you can see an improvement in the rating scales for their Parkinson's. So this two sets of data was then used to get the funding from the EU for the FAIR-Parks 2 study. So these were studies done in people with reasonably advanced Parkinson's, and they were on standard Parkinson's medication, and that's important. So the next study, which was done in about 372 patients, so 186 were assigned to receive deferoprone and a matched number of placebos. So these were done in very early Parkinson's cases, so they'd just been diagnosed, but they weren't receiving any therapy. And to our surprise, it actually made the Parkinson's worse. So this really wasn't the news that we were expecting. So if you look at the change from the baseline, so in the placebo it was 6.3 worsening. So you might expect that over 36 weeks. But in the deferoprone treated individuals, it was actually 15.6, so it's almost 10 points worsening over the placebo. Why was this really happening? It's really, you need to think about the group of patients that were treated with deferoprone. So tyrosine hydroxylase is the rate limiting enzyme in dopamine synthesis. So here you can see the structure. You've got iron at the catalytic core there, and so this plays a key role in the synthesis of dopamine. So people who have early stages of Parkinson's, their remaining neurons are going into overdrive. They need to produce all of the dopamine to keep people moving, and so they're heavily reliant on the tyrosine hydroxylase. And the chelator must have been taking the iron out of the catalytic core. Now, in our animal models, we showed that this only happened at very high doses, so at above 100 mg per kg, when we were actually dosing with deferoprone in humans at 30 mg per kg. It may be that since they're heavily reliant on tyrosine hydroxylase, is that it was taking some of the iron out, so making them that they couldn't synthesize dopamine, so it appeared to be making the condition worse. Whereas in the L-DOPA treated individuals, you can see that the L-DOPA, if you give it orally as a drug, it bypasses this step altogether, so you get continuous production of dopamine. So in those individuals, you didn't see any changes. So this was really supported via the prolactin data, because if you know your biology, that dopamine is also produced and serves as an inhibitory transmitter for prolactin synthesis. And so the dopamine production here was less, and so there was an increase in prolactin levels. So this, it's still in a quandary about whether we go on and do another trial in L-DOPA treated patients. And so what I'll do is I'll draw that to a close there and answer any questions. So as you can see, there's a lot of drug development within the field at the moment, so it is quite an exciting time for people affected by Parkinson's.
Questions?
Yes, right at the beginning you suggested that the vagus nerve might be explanation as to why these people are getting Parkinson's disease. What is it in the vagus nerve that is enhancing the inflammation, etc., in the substantia nigra? Is that something worthy of looking at? Trying to stop it?
Well, it's probably in the Parkinson's gut. There is they start, rather than having a normal balanced flora of bacteria, they tend to have a higher amount of pro-inflammatory bacteria in the gut. And And because you've got the nerve endings from the vagus stimulating gut movement, you've got the alpha-synuclein there in the vesicular trafficking. This inflammatory activity may cause the alpha-synuclein that's in those nerve endings to misfold. And then what it can do is start to track back up the fibres, and then it goes to the dorsal motor nucleus there. And so then it can actually be, released again within that area, taken up by the next neurons and go further up the J. So it's this propagation system that's been hypothesized. And so one of the areas where this could start is in the gut. So with these,
knock out the inflammatory bacteria in the gut, because you seem to know what they are.
well, we don't know exactly. So there's been multiple types implicated.
Mm-hmm.
There's only been some recent studies where they've identified some key species. But then, even as you know, the antibiotics are going to be quite indiscriminate. So if you actually treat with antibiotics, you're going to wipe out some of the good ones as well as some of the bad ones. So what some people have been proposing is whether you actually do, fecal transplants, so that you actually put in a new population. So wipe out everything and then put a new set of bacteria in there, which mirrors more what you see in the human. So they've actually taken fecal material out of the guts of people with Parkinson's and put it in the animals that overexpress alpha-synuclein, and you actually see transmission from the gut going to the brain. So there is some support for that, but there's still a lot more work to do before we can actually say, we should be treating everybody with antibiotics. And also, from the brain bank that we did a study, not everybody follows that route. It's about 76% of people have the early brainstem degeneration, about 26% didn't. And so we think it might be an environmental route through the olfactory bulb, which is again, you see lots of misfolded alpha-synuclein in the olfactory bulb, and that could be a route into the brain as well.
Yes, please. Alec?
I'm probably continuing Bobby's question, and I'm going to give a short introduction to the question. First of all, a very enlightening talk. Thank you very much for that. 2 weeks ago, I was in a meeting in Switzerland. I heard Emily Balskush from Harvard talk about the gut microbiome and what they have been doing. And they have, for instance, they have done something completely unique, I think. They have been able to figure out the structure of the toxins secreted by Clostridium, by the Colibactin, by non-chemical means, by genetic means to identify the chemical structure of the toxin. Now, about the same time, I think it was the same week when I read from the papers that the desulfovibrio bacteria have again become prominent in Parkinson's. And I was wondering whether there's any activity, do you know if there's any activity about looking at the human gut microbiome and what might be the causative agent, perhaps?
So we don't know why the gut flora changes, and also we don't know, because also quite a lot of people that were tested where they looked at their gut flora, they were already on dopaminergic treatment, and also in Parkinson's you have constipation. So you've got a lot of factors which could influence that. So now we can pick up people years before they start clinical symptoms and before they start therapy. We could actually look at the microbiome there. But also a lot of these pro-inflammatory bacteria produce a protein called curly. And again, this causes sort of amyloidogenic proteins, to increase their propensity to misfold. So there are some approaches which are trying to target this curly protein that's actually produced by some of these bacteria.
I'm interested, earlier in the presentation you talked about some development in biomarkers and diagnosis. And you mentioned that with a combination of tests you could be within 98% effective. But at what stage in the Parkinson's development are you talking about in terms of the detectability? Are you talking about at the stage where you've got the brainstem effects, or later?
Yeah, so this is going to be very early. So in order to do the seeding assay, so you're just going to need bits of bad protein within your cerebrospinal fluid. So there it could have reached the lower part of the brainstem, and then the cell realizes it's toxic, starts to kick it out.
Right.
So it'll actually end up, some of it will in the CSF.
Yeah.
So the idea is we're starting now with the Michael J. Fox Foundation. We're starting the next phase of their recruitment study. So this brings in the potential of prevention studies for the first time, and hence my title really. So we don't know how far back we can go, and we don't know how long it will take to go from one stage to another, but we think we can go back, quite far in the stage. So what you can do is you can pick up people who are seeding positive, but when you do a DAT scan, a brain scan, it's normal. So the level of the alpha-synuclein hasn't got bad enough to start causing death of dopaminergic neurons. Or you can do a brain scan and they're intermediate from a healthy control and somebody with Parkinson's. So there you've picked people up who are on the way to developing dopaminergic degeneration. So you should be able to pick people up at different stages, and then you should also be able to use those biomarkers as a progression. So if you can pick people up at just seeding positive, DAT scan negative, if you can prevent them progressing to a DAT scan deficit, that would be a major.
That's something.
Yeah.
A follow-up question. I mean, you didn't mention it in the presentation because I'm sure there's many, many other developments going on. But in the press and BBC was several programs about a lady who can smell people with Parkinson's.
Yes.
And I just wonder whether they've been successful in Manchester, where they've identified markers, real markers.
Yeah, so Parkinson's UK actually funded the first part of that study.
Yeah.
So we had it brought to us and we thought it sounded a bit strange.
A bit mad.
So we funded, I think it was 20 normal white t-shirts that went out. People wore them for 2 weeks on and off, didn't wear them all the time, but then they were cut in half, they were sealed in bags, and then she unwrapped, smelt them, and actually said whether it was somebody from, it was a healthy donor, or whether it was somebody who had Parkinson's. And she guessed them all correct, apart from one person, which was actually somebody who was a healthy person. But then 6 months later, that person actually developed Parkinson's.
Yeah, this is fascinating. But so that's quite an advanced stage that they were at.
Yeah. So now they've actually done other studies where she's been able to distinguish the smell at different stages, and so the group in Manchester have actually now it's moved to swabbing the back, because that is where there is a lot of sebum production, and so the changes in smell that she's detecting is a change in sebum, and there are three distinct chemical changes within the sebum that are very characteristic and very highly predictive. So they're just developing that into a biomarker test at the moment.
Right. And these biomarkers how will they correlate to this early diagnostic that you...
Yeah, well that's it. So to do we really need to do the head-to-head studies.
Right.
So, can she pick people up when they're seeding positive, or is she picking people up when they're starting to see some degeneration in the DAT scan?
I mean, just for the benefit of everybody else, so that the piece that was most publicized was the piece where she reckoned that her husband,
that's where it came from.
He started to smell bad to her 10 years before he was diagnosed.
Yeah. So then Parkinson's UK has support groups all over the whole of the UK, and so her husband went along to one of the Parkinson's branch meetings, and she said to her husband, "They all smell like you do." And everybody thought she was just crazy. And when it comes to her family, her mother and her sister, they're super smellers. They're people that are used in the perfume industry. And she could actually smell the differences between the people in the room that had Parkinson's and that didn't. And often, sometimes when I meet up with her, I'm thinking, how close should I be standing? Yeah, it's a really interesting story. Now it's going, to be developed as a full-blown biomarker. So there have been, we did write a letter of support. They were trying to get one of those like biofilms that you could actually stick on the skin and to see whether you could actually do it by a biofilm.
There's pictures of her hooked up to a gas chromatograph.
That's right, yeah. So from the test, from the sebum swab, it goes through the machine. She gets half of it and the machine gets the other half. And she will actually grade the severity of their Parkinson's just from her smell. And then they'll look at what the gas chromatogram is actually coming out with the readings as well. So we don't know whether it's going to be something that you could use in progression monitoring as well.
Yeah.
So, interesting. Yeah.
Other questions?
Yeah, I have another one. So I'm fascinated by the iron connection.
Yes.
Okay, and only because I've got a near relative who has suffered for many years of hemochromatosis,
Yes.
which is an overload of iron in the brain. Which
It's in the periphery.
well, yeah, but it manifested itself as in neurological symptoms,
Oh, right, okay.
and he had a lot of very similar symptoms to Parkinson's. And then his treatment was mostly just to bleed him.
That's the only
Yes,
treatment
that's
for
right,
hemochromatosis.
Yeah,
There's no point giving them iron for
he just fed the roses every month.
It's rapidly capable of replacing the blood, the hemoglobin from the reserves,
Yeah.
because they've got buckets of reserves of iron. So the best way is to venous section.
It's got very healthy roses.
Anymore.
Yes, it's a fascinating thing.
There are some strange studies about hemochromatosis
Oh, yeah.
and neurodegenerative disease in general, which suggest that the correlation is not there at all.
No. Because they've looked at quite a few cases of hemochromatosis and done the MRI scanning, and it doesn't seem to correlate into the brain.
Well, on a sample of one, we can tell you there really are. Because after 20 years of being bled, his neurological symptoms very, 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 very slowly declined until it doesn't have them anymore.
Yeah, the distribution of the iron is important as well, not just. This is one of the first things that we learned when we started working on brain iron. There's no use just doing total brain iron. You need to actually, that's why we went to David, to dissect out parts of the brain and know that we're looking at the brain in specific brain areas. Because just looking at total brain iron doesn't really tell you anything.
Sure, he takes students.
Anyway, I think we can move to the more informal discussion over drinks and nibbles. And before we do that, I'm going to give you something that's not our usual liquid present. It's still a liquid present, but it's chosen as a function of your preference.
Yes,
Gin rather than whiskey.
That is my preference. Thank you very much. Yeah.
And
More of a
thank you
gin
once
drink.
Please feel free to interact with the speaker during the informal break. Thank you all.
Take that.